I'm Laura Bonnell, and this is the Living with Cystic Fibrosis podcast coming to you from Detroit. Please subscribe and even rate and comment on our podcasts when it moves you. I learn something new with every podcast, and I'm always inspired by the people that are showcased here. We don't give medical advice. You need to connect with your doctor for that. I hope that this Living with Cystic Fibrosis podcast educates you and sparks some volunteering or advocacy. The CF community needs people like you. Thanks to our sponsors, Viatris and Vertex, for their support. I do say this a lot when I have scientists on the podcast. Scientists are some of the coolest, smartest, funniest people that I know, and I am excited to share what they're doing in terms that you can understand. And Dr. Tom Kaiser is no exception. He lives and works in Durham, North Carolina, and he's a wonderful mix of scientist, physician, and entrepreneur who is focused on designing better medicines using cutting-edge technology. And he began his career at Emory University in Dennis Leota's lab, where he worked on antiviral drug discovery and later helped pioneer early machine learning approaches in drug design. So it's very interesting and also exciting. His work spans areas like RSV, cancer, and neurodegenerative diseases. And he went on to earn his medical degree from the University of Oxford. He is also the co-founder, the chief scientific officer of Avicenna Biosciences. He's leading the development of innovative therapies aimed at improving and saving lives. And he has an interesting story about how he named the company. He ends his bio by mentioning the love of his life, his wife, and we will hear about that as well and their relationship. And I'm just going to begin this podcast by saying that what I told Dr. Kaiser that I thought that, wow, this guy is so cool. He must be in a movie. And we met in DC during a Mish Bio fly-in. And so it's always fun to meet somebody in person and then do the podcast, because you get to know them a little bit. So, you're in for some really interesting conversation, and you're going to be a big fan as soon as you hear more from Dr. Tom Kaiser. Dr. Kaiser, welcome to the Living with Cystic Fibrosis podcast. Excited for everyone to meet you. So thanks for joining us.
Yeah, thank you for having me. It's a pleasure to connect on some rare disease topics, and it was good to meet you last week in DC.
Yeah, it was, and I am excited for everyone to get to know what you're doing. And first of all, tell us the story of kind of how you got into science. Was it something as a kid you thought that this is where the direction you wanted to head to, or how did that go?
Yeah, I think it definitely was when I was a kid. Actually, when we were moving into our new house, I had some knickknacks from my elementary school that I hadn't looked at in a couple of years. And in the little author bio, this little fake publishing house that we had, I think in the 5th grade, it says little Tommy wants to be a scientist. So it's been that way for a long time. And I just thought, as a kid, these incredible people on the vanguard, pushing the frontier of human knowledge, turning things that used to be considered impossible into practical everyday realities. I mean, just think about the routine nature of flying, right? 1,000 years ago, that would be something that only divine beings did. Now we're bothered by airplane delays. So as a kid, I was very interested in mathematics. And then a family physician, our family friend who was also a physician, said, you should really look at medicine. You should think about doing that for a career. And I thought, I don't know. So he took me into his clinic. He's an OB-GYN. And I just saw some incredible things. The first day I went in with him was actually a surgical day. And I thought, I didn't know you could do this for a living. This is amazing. And so I thought, I still want to be doing mathematics. I'll keep that as a hobby. So I went to undergrad in biochemistry. And from there, as you get into organic chemistry, I fell in love with it. I thought it was incredible that you could You could make molecules that cured illness. You could make molecules that altered the perception of reality. You could make molecules that were horrific weapons. It seemed like such a powerful discipline. And then I got into medicinal chemistry and I was hooked. I wanted to go and design drugs. So the rest of my training really revolved around that. I trained in synthetic organic chemistry, organometallics, then medicinal chemistry, and then I actually went back and qualified in medicine over in the UK. So it's kind of been a flurry of multidisciplinary interests, trying to put mathematics, chemistry, and medicine together in a powerful combination to discover the next generation of therapeutics.
And it is so cool. And I have to say, like, I knew I wanted to be a news reporter way back when I was younger. And I remember in first grade, Dr. Berger was his name, and he gave me this award that said I was going to be a journalist. And I just think, I mean, so many people knew what they wanted to do when they were young, and we followed our dreams. Isn't that just so? Aren't you proud of that you followed the direction, and look at you? I mean, successful and really and truly, like they say, living the dream. I mean, I think it's so very cool.
I am very proud of what it took to get here, and very proud of the kind of work that we do at Avicenna. But yeah, it is a great privilege. I mean, we get to be on the frontier on a daily basis, discovering new things, seeing new horizons. And I think that's incredible. It's an immense privilege. And that I wanted to do it as a child, and I get to do it professionally now. I mean, it is living a dream. Absolutely.
It is. And we're going to get to the advocacy in a little bit, but I wanted to also go to I'm not sure if we head to the UK where you were first, or the serendipitous story that you told me in DC. Can you tell us how it kind of tied you into what you're doing now?
I'll give the story of my postdoc, which I was brought on by a man named Dennis Leota, who is probably the most successful academic medicinal chemist, if not one of the most successful. The amazing thing about the Leota group is it's chock full of brilliant people who are very accomplished in medicinal chemistry, drug design. And Dennis himself has put out something, I think it's getting close to 30 new medicines now. So combinations of molecules that were discovered in his lab, and even founding companies that would go on to cure previously uncurable diseases. So Pharmacet, sofosbuvir, the cure for HCV, came out of a company that Dennis founded. So it's an extraordinary place to go and learn medicinal chemistry. And I was very fortunate that I was put in charge of the nucleoscience group, which is kind of the new chemistry group for antivirals. So we're doing new chemistry, oftentimes going after some novel biological angle in a virus. The issue with that is that because you're doing new chemistry, you don't know what you don't know. And so there's a lot of new reactions to discover, a lot of new stereochemical insights, like the nuances of the three-dimensional molecular landscape that you have to traverse. And as a consequence of that, there's just immense failure. Like, we're really good at spending a lot of time and effort to discover some really new chemistry that isn't practically useful. And I thought, well, I want to design some medicines. I don't know that I want to be on the pure chemistry side. And so Dennis gave me a huge amount of latitude. So there were some people that were playing with what would become machine learning, but it was called automated QSAR. This is like back in 2012, 2013. So way before AI was a buzzword. And they were working on potency optimization, which is one of the key tasks of a drug, trying to make it very avid and selective for whatever macromolecule you're trying to modulate. So the target, right? The way a drug works. And I thought, well, this seems really cool. Let's test it out prospectively. Let's see how it works. And we found that we were really good at optimizing chemical space, and eventually we became very good at optimizing what's called a lead. So the first time you really get animal efficacy. But beyond that, going towards clinical candidates, we still had some limitations. And so I started building some technology that would eventually become the heart of Avicenna. And at this time, I had decided to go back to medical school And so I actually went over to the UK to study a single payer healthcare system. That's why I went to Britain. And so while I was in the UK, my co-founder and I, we were looking for ways to keep our academic careers moving forward because he wasn't tenure track yet. And I was a medical student, so nobody's going to give us grant money. So we formed a company to raise some SBIR funding. And before we ever actually submitted our first grant, a venture capital group named DCVC Bio came along and they said, "Hey, what you guys doing is really cool. Do you want some money?" We said, "Sure, yeah, we'll take some money."
Absolutely, right? How much? Yeah.
Yeah. And so we raised our pre-seed at $2.5 million to prove that we could actually find a lead-like compound with basically the flick of a wrist or a snap of our finger. And more importantly, the thing that I love about DCVC Bio is they brought in a lot of know-how, right? These are people that have built a lot of successful companies. Chris Meldrum joined us as an entrepreneur in residence. He knows how to run a successful biotech. And so that operational insight with research funding allowed us to very quickly get 2 new programs moving forward, one of which was in ALS. And 11 compounds, we found what would become our clinical candidate, Avicenna. So we made only 11 molecules, and out of those 2 were clinical candidates. So the technology worked exactly as we said it would.
What was that day like?
It was a good day, to put it mildly. And I think that's one of the crazy things about that story is that the data package that proves you have a candidate takes years to make and millions of dollars. And so it wasn't really a day. It was more a series of years of, wow, this compound is really shining. It's highly CNS penetrant. It's very efficacious in animal models of neurodegeneration. And it's very selective and should have a good safety profile. So the feeling that you can indeed basically flick a light switch and find a new clinical candidate using some machine learning that we developed, that's a good feeling.
It's incredible. And were you going to explain more about that? Was it also Parkinson's or this PPMS, which I don't recall what that stands for? I'm sorry.
Primary progressive MS. No, this is for ALS.
Okay.
The program is working on aspects of Parkinson's disease as well. So really going after, you can kind of think of it as a hijacked stress response. So there are things that the cell does to recover from oxidative stress or even mechanical stress. And as long as that's in the short term, that's healthy. That's part of the wound healing response. But when that becomes constitutively active, it's disastrous. You destroy the orderly tissue. Your innate immune system comes in. It starts destroying healthy tissue alongside the diseased tissue. And that's just disastrous. And that's one of the reasons why ALS progresses so quickly, is that you have that infiltration of what are called microglial cells, and they just destroy healthy axonal projections and neuron bodies in the spinal cord and motor cortex. So it's very aggressive. It's very nasty. And the cool thing about our molecule is it blocks multiple processes at the same time in a selective fashion. And that's kind of the theme that we take to rare diseases, is interrupting that disease process in a targeted fashion, often with multiple angles of attack.
Can you explain a little bit more what you're doing, what this means for ALS, or some people, I guess, that have ALS? Yeah.
Yeah, we're actually going after disease modification. So we want to add years of life to people with ALS. So actually interrupting the disease process and slowing it down and giving patients ideally years of life with their loved ones, where they may have only 6 months now, maybe 2 or 3 years. And it's part of a concerted effort on our part of going after ALS from a variety of different choke points from the pathology. So we have other ALS programs that are earlier, but really trying to come up with multimodal means of arresting that disease and stopping it in its tracks.
And so, like cystic fibrosis, are there different variants of, there are different variants of ALS, obviously, but so what you're doing, will it impact all forms of ALS or only certain?
Yeah, so right now we're working on fast progressive ALS. So the really accelerated decline and deterioration that you see in some variants of ALS, it seems like our 2 targets are ROC1 and ROC2. There's ample human clinical evidence that these two targets are really involved in fast progressing ALS. But I think that's one of the interesting things about rare diseases is oftentimes it's treated as if it's a universal syndrome, but there are a lot of subvariants, and there are heritable forms. This is not so true for cystic fibrosis because there's always a heritable form, but with something like ALS or primary progressive MS or systemic sclerosis, there are rare heritable variants, but most of the time it's a sporadic disease. And so what that points to is you actually need this pathway disruption or the system disruption approach, because these single targets, well, it might be sufficient to cause ALS in those heritable variants, but it's not necessary, right? There are other kinds of dysfunction that give rise to something that looks very similar clinically. And so picking that apart and being very deliberate about which kinds of subvariants you're going to go after and how you're going to approach that, something that we're really mindful about at Avicenna.
You must be talking to so many patients and people who have loved ones who have ALS, because I know a friend of mine's husband who has ALS, and as everyone knows, he's gone from standing to not being able to hug people to now in a wheelchair, and it is going so fast. So, how many, is it years away before you get to where even to clinical trials? Or
we're submitting our pre-IND letter right now. So we're in the last preclinical phase, and the hope is that we'll be in the clinic next year.
Wow.
Do a healthy volunteer study, make sure that we have good safety, and we get to the central compartments that we need to get into. But the hope is that we can bring this therapy to ALS patients in the short term.
So a year from now, people could be in the clinical trials?
Yeah, but these would be healthy volunteers. So
Okay.
it'd probably be 2 years, 2.5 years before we start our phase 2. So we're close.
So exciting for the whole community, regardless, I think, of where people are, because you're just so connected to the community. Even if a loved one has it and they can't be helped, you want this hope for future people that are, that have ALS. But when you get to the clinical trials, I have so many questions, because everybody I talk to in the CF community, as far as therapeutics or whomever, for whatever reason, sometimes it is hard to get people in clinical trials. I know there's so many different reasons, but do you think you're going to have any challenges getting people in clinical trials? And then do you have any idea yet how many people you'll need?
We have a pretty clear idea of how many people we would need. It'll probably be somewhere around 150, 180 patients.
Wow.
So fairly small clinical trial. And I think that's part of what one of the strengths of our approach is. We've been very mindful to partner with some major players in the space. So a gentleman named Jonathan Glass, he's a neurologist at Emory. He's a major trialist in the ALS space. He serves as a member of the scientific advisory board for Avicenna. We have a very close relationship with ALS TDI, the largest nonprofit research group dedicated to curing ALS. So we really made sure that we have good connections with some major pillars of the community so that when we're ready to move, we can move quickly.
And that is so exciting. And so it's down the road, but is there a way that people can follow what you're doing now? So when that clinical trial comes out, I'm sure, you say it's a small study, but I think it's so exciting. 150 people sounds incredible to me. And I do realize it's small, but it's still so exciting. How can people stay in touch so they know what you're doing?
Avicenna posts on LinkedIn. So if you want to follow our progress, you can just click on the follow button for Avicenna and get an update for as we move through the IND process and eventually through early clinical trials. We'll make press releases about that. And
we have the link in our show notes. So if people don't know how to spell it, it's in there. So just look at the description in show notes and people can click on it and get right to you then. And tell us about the name, how that came about.
Yeah, so it's actually one of my childhood heroes. And so Avicenna is a Latinization of a Persian polymath who lived about 1,000 years ago. His name was Ibn Sina, and he was a physician. He was an alchemist, a mathematician, a theologian, a legal scholar. He was a real Renaissance man before the Renaissance, right? That's one of the interesting things about the Islamic Golden Ages. It was the Renaissance of the Middle East, this huge intellectual revolution. And I thought it was really appropriate for what we were trying to do, because we're trying to put mathematics, chemistry, and medicine together to find impactful therapies. And I thought, well, if ever there were a mascot, it would be Ibn Sina. So we called ourselves Avicenna. But yeah, he wrote a book called The Canon of Medicine, and it defined medical thinking for about 600 years. So it's
Wow.
major intellectual in humanity's past.
That is really cool. Just the meaning alone is very cool. How do you feel the future of rare disease is going, and the future of science right now?
Yeah. How do I feel it's going? Rough. I think that's why we were on Capitol Hill last week, right? Is to
Right.
get the message out that we need stability. We're trying to work on very high risk endeavors over the course of a decade. And if there's regulatory uncertainty, financial uncertainty, then these new drugs never reach the people that need them. That would be a tragedy. So I think the community, the scientific community, both academic and industrial, is unified in that we need to keep funding moving forward, to keep the lights on so that we can build a brighter future. And I think that's especially true in rare diseases because they're considered very high risk investments, right? Oftentimes they're poorly understood because they're not commonly studied. And so you're trying to breathe a spark of life into a program that requires a lot of money. And investors might look at that with a raised eyebrow because they view it as very risky. So we need more stability so that we can keep on going after these great unsolved medical mysteries and turn them into things that are treatable.
And how did you feel your time in DC was, this is all bipartisan stuff we're talking about to lawmakers. And I did feel like both sides were listening. So I am hopeful. And then the SBIR that you were talking about earlier, it did pass. Yay,
Absolutely.
so our efforts did pay off because we were in DC last year talking about the need for that to pass. So how did you feel after those couple of days in DC advocating?
I felt very good. I felt heard. And it was great to be a part of the North Carolina contingent. So Laura Gunter has really put together an excellent infrastructure, and she works very closely with the delegation from North Carolina to make sure that real important policy measures that help biotech, not only in North Carolina but in the United States, are put forward and advocated for. So a great example of this is the EPIC Act, right? So removing the small molecule pill penalty. That is something that Laura's really been putting her weight behind. So people know how important this is. They know that it's a very difficult enterprise, that there needs to be stability, but they don't know exactly what they need to do. And so helping them understand specific policies that can help out us trying to discover new medicines, I thought was very good conversation. And then also just going and educating and teaching in general. There were some members of Congress that didn't know why the NIH or NSF funding was so important. They thought, well, couldn't these scientists just go and get other jobs? And most of the NIH and NSF funding goes to academic groups. Not only do we get knowledge from that, but we get the next generation of scientists, right? So the federal government pays for scientists from around the world to come here and get their advanced training. In exchange for that, the United States gets research that it considers of strategic importance. And some members of Congress didn't know that. So
Wow.
I thought helping them understand that it's more than just jobs per se. It's actually training the next generation of scientists. That it's vital that we do that. Otherwise, we'll lose our competitive advantage.
Exactly. And I think too, just from the Michigan perspective of cystic fibrosis, there are so many wonderful, older, very experienced doctors, but getting the fellows in is harder. Getting them, to Michigan to learn and then to stay. Our CF clinics here really need to have a really good balance so that there aren't any gaps when some people retire. So I know the clinics here are working on that for that very reason. It's very interesting to go and be an advocate, isn't it? To go to DC.
Absolutely. Yeah. It's always different. This was my, I think, fourth time being on the Hill. What's interesting is the energy is always high, but it's always a different kind of energy. There's usually something that's being negotiated while you're there, or there's some voting session or whatever that's just very turbulent. It's fun, and then you come home and you realize, oh, I need a day to decompress from that.
Oh my gosh, I was dehydrated, starving. After those 2 days, it felt like a lot more days. So, and you're just zooming around, walking so fast from one office to the next. But so exciting and so fun. And then you meet so many great people, other advocates too. What did you take away as far as something that you thought, wow, that really impacted me? It's moving me forward, or it's keeping me really excited and feeling the energy of advocacy.
Yeah, I think just knowing that there's so many people that are aligned in making this work, right? Sometimes when you're working on your own endeavors, you feel isolated. You feel like the world is against you just because it's so hard. But going out and seeing that there is a community, that we're all trying to bring the next generation of medicines into reality together is such a great feeling.
And, stealing scientists is nothing I should advocate, but wow, it'd be great to have you in the CF community is all I can say. But I wouldn't do that to ALS or any disease. And it's also important to say that what you do in ALS or whatever you're doing will impact the rare community, and it will impact cystic fibrosis because everybody's learning from everyone else, right?
Yeah, I think that's been one of the big things of the past really 6 or so years is people are trying to accelerate clinical development in rare diseases, and it's not working. And I think we really ought to heed some of these lessons. A lot of these rare diseases are multifactorial. They take years to decades to show up. So there's a lot of disease momentum there. And I think trying to get signal in 6, 8 weeks in a phase 1b, we might be throwing away some good therapeutics. So I think we're starting to get insight into just rare diseases in general. How do we improve diagnostic categories so that we can segment the subvariants of a rare disease? And then also, how do we really run an appropriate proof of concept clinical trial? And maybe we go with a pivotal study, a little bit of a larger phase 2 study, so that we can shorten the time to patients if it works in a proper clinical trial. And I think these are some of the lessons that ALS is facing in terms of digesting, certainly primary progressive multiple sclerosis, another rare disease that we work on. There is some thinking there about how to improve clinical trial design. But I think that's going to be one of the things moving forward is maybe we ought to run a more, what I would say, classical clinical development program so that we don't throw away promising therapies because we don't see efficacy in phase 1b.
I also think it's important that people know that, and this is one of the things we talked about in DC as well, as we were talking to lawmakers, is that everyone's not a huge pharmaceutical company or a huge therapeutics company, right? I mean, I don't know if it's fair to say that you're a small company, but the smaller companies I've met with have great ideas. Like, this is the next thing. This is what you are all doing it's going to be big, it's going to help so many people. And I guess what I'm trying to say is it's just a reminder to people that whatever you do, it has an impact,
Yeah,
and it can grow into, I don't know, a cure or a clinical trial or whatever it is, right?
absolutely. We're a group, a very small team of people, 12 people, and it's incredible to go to work with these people because they're willing to work very hard, often make a lower salary. That's the big lever that you have with a biotech is, well, there's equity, but most of the time that doesn't work out, right? So these people are willing to expend their talent because they believe in something greater, something that could take away another's suffering. And they're really willing to go through Herculean efforts to make that possible. And it's the tiny teams at the very beginning that design the future of medicine. And it's a great privilege to work with some of these exceptional people. But I think we tabulated it at Avicenna. Most of the people that work at Avicenna are naturalized American citizens. So the best and the brightest from
Wow.
around the world come to the U.S. and we get to benefit of their intellectual labor. And I think it's very important that we protect that, we keep that, because that's one of the things that makes this country so strong is the diversity of talent that comes here in search of a better life and in search of a new horizon.
I'm so glad you said that. That is the most beautiful thing out of the entire podcast, or one of the most beautiful, because I felt that way in DC, or I always feel when there's so many different people speaking to lawmakers, and you have a diverse group, or even at our education day, a diverse group, or when I travel to Egypt, and I just think fear is a scary thing. And I mean, what I mean by that is people who are afraid of another culture, or people who are afraid because of how someone looks, or it's just such a waste of time and energy and brilliance. Like, we have so many cool people. So anyways, the long-winded thing I'm saying is I love that you said that. It's so important. And to, like you said, to keep that genius and that energy, you know, in the US, and being open to so many wonderful, bright people.
Absolutely.
Is there anything else, I'm sure there is, that you are working on or thinking about for the future of either the rare community or just science in general?
We really work on those rare diseases that have an autoimmune component, but specifically an innate immune dysfunction. And so that's one of the fun things about what we're doing is trying to disrupt fibrosis, maladaptive wound healing. So there are a variety of different conditions that all have that hallmark. And in fact, if you look at amyotrophic lateral sclerosis or multiple sclerosis or systemic sclerosis, you can see a theme, right? That we're going after this common nexus of pathology in our indications. But I think just making sure that we can develop the therapies that come spilling out of our platform, that's really the priority, and then continue to refine what we're doing on the machine learning front. We found some really interesting things in terms of how to navigate the nuances of data sets. We call this inferential mechanics. So the ability to find hidden mechanisms in a data set that you otherwise believe is uniform, and how that impacts clinical candidate design and efficiencies of clinical candidate design. So just balancing those two areas of research, the pure math ML side of things, and then the practical medicinal chemistry, pharmacology, drug development side of things. I think that's fun. That's enough to keep me busy for a few decades.
Right? Yeah, that is great. Yes, please stay busy because we appreciate it. And what does your wife think? I don't know. She could be a scientist too. I'm not sure. But what does she think? Does she appreciate what you do? And then the other thing I always say to scientists or the pulmonologists is, like, do you have family time? Because you're constantly working on this disease.
Yeah, I mean that's been so my wife and I will have been married 18 years this year, and I was married when I was 22 years old. So you know, when you see what you want, you can put it doesn't take much difficulty to make a devotion to that. And just thinking about the last almost 20 years, I mean, she's been with me through all of my training and then all of
Wow.
getting Avicenna going. So it's been intense. And I think we're just now settling into some semblance of a normal life where we might have a little bit of time on the weekends, or we can take a weekend trip somewhere away from our house. And I think that's good, because you really do have to balance a shared life with your partner versus the high intensity research endeavors that consume you, and they will consume you if you let them. I think that's really important to talk about. Is it's not just me who pays the price, right? It's my wife, where I'm away for weeks or months doing something. Like when I was in the UK finishing out my medical training, this was in the midst of COVID. So I would be deployed to rural hospitals in England for weeks to months. And she was stuck at our home all by herself under lockdown. So it really has been an intense series of decades to get here, but we've had a good time together. So I think that's really important, is finding the things that you can celebrate, finding the things that can make you happy.
Well said. And I think that it's so important to think of our spouses or our partners or whomever it is, whatever side you're on, be it the scientist or the caregiver's side, because they are directly impacted by everything that's going on. And to new or potential scientists coming up, kids deciding what they want to do in college, what do you say to them? I mean, I would think that they would be a lot like us. They probably have known it or felt it when they were in elementary school or at some point in, K through 12. What do you say to them as they try to figure out their future careers?
Just do it for fun. Do it because you're curious. Don't worry about the end result. Just be mindful of why you got into science or why you got into medicine, is that it's important to you and that it is fun. There is always some great joy in the act of creation for a new idea, and that will carry you through the hard parts when you're filled with doubt, when the failure is all around you. How do you push through that? The answer is you push through it because you want to, and because you like to find creative solutions to problems that you don't know how to solve today, but you might solve tomorrow.
And that is a good point about failure. I think we try and push it under the rug or never attest to it sometimes. And it's so critically important to our future, right?
Yeah, if you're doing research right, a failure means you learned something about the universe, right? And it was a good idea at the time, but now we know more. One of the most important things about being a good scientist is being flexible with what you believe to be true and doing a good experiment in a very high quality manner and actually learning from those data that come back. You're being taught something by your experiment. And so how do you adapt to that? Is it recoverable? Do I need to change the question that I'm asking, or am I asking the wrong questions entirely? Right. That part of being a scientist is really where it becomes an art, is how do I approach this nebulous answer that is not quite what I thought it was going to be, but also sort of kind of what I thought it was going to be. Like, how do I make sense of that? Right. And that I think is the real art of science, is how do you push forward with nebulous data where you can actually build towards something that's concrete, tangible, and then take the next step.
And I'm sure you've written a ton of scientific stuff, but have you ever written a book? Do you think about it? I think if you wrote a book, it would be a great book. It would you would weave in so many cool thoughts and things. Do you think about doing anything like that?
Uh, maybe, maybe. I tend to be a fairly shy person. I know you probably don't believe me, but Maybe when I have when I have a couple of medicines out in the clinic, maybe I'll talk about the ones that didn't come to the clinic.
Oh, that would be interesting, though. That would be really interesting because you find out about how you push forward and how you get to where you are and all of that. The journey, right? What we always talk
Absolutely.
about. Anything else that you want to share with us before we wrap up?
I think it's just important to talk about advocacy and how it takes many forms. I think that's one of the things that I love about the scientific community is you'll often find people who are willing to go, especially to elementary schools, junior highs, high schools, and get children excited about science, that they can believe that they can do science too. I think that's very important. So advocacy from elementary school to Congress, I think that's where we are right now, is we need to instill in our fellow citizens a sense of wonder, a sense of curiosity, and a sense of erudition so that we can continue to tackle the world's problems.
I agree. And it's funny because yesterday I wrote down in my notebook here, write a book about advocacy and the journey, just because, I mean, you've been doing it so long, I've been doing it so long, and it empowers you, right? It uplifts you so much, regardless, even if you have someone who's like not interested in what you're saying, clearly not interested. I mean, most of them are, but every once in a while. And it's just a challenge then. How do I get them to hear me? Or how do I get them to understand the importance of it? So, yeah, advocacy is so important and powerful. It's how we've changed the U.S. all these years and how we keep going.
Absolutely.
Well, I wish everyone could meet you. You are very cool. And I hope you do write a book because I think you have a lot to say and you're having a huge impact in the U.S. already. So thank you for everything you're doing.
Thank you, Laura. It was great to meet you. And I look forward to many conversations over the years about how we can advocate.
Absolutely. Thank you, Dr. Tom Kaiser. The original music in this podcast is performed by Kevin Allen.
It's not complicated.
Who happens to have cystic fibrosis.
We all got our worries and fears. I know it's got you frustrated, but loving you is so alright.
This has been the Living with Cystic Fibrosis podcast. For more information and to learn more about the Bonnell Foundation, visit our website at thebonnellfoundation.org. That's the BONNELL Foundation.org. This podcast was sponsored by Vertex Pharmaceutical, The Science of Possibility, and Viatris. It was produced by Jag and Detroit Podcasts. Follow our show in Apple Podcasts, Spotify, or wherever you're listening right now.